Everyone, myself Avanti Pandey, and our today's topic is ion exchange chromatography. Now let's just start with an introductory portion. The principle of ion exchange chromatography is based upon the simple fact that different cations or anions have different capacity to undergo exchange reaction on surface of a given exchanger. Ion exchange chromatography is form of adsorption chromatography. Now what is adsorption chromatography? So it is nothing but the process of separating chemical by means of interaction between adsorbent and adsorbate. As we are concerned with ion exchange chromatography, we have to know what is ions. Ion is nothing but a charged molecule. So it may be negative or may be positive. So ion exchange chromatography, it has two type of exchangers, that is cationic exchanger and anionic exchanger. Cationic exchanger possesses negatively charged group and this will attract positively charged cations. These exchanger are also called as acidic ion exchange materials because their negative charge result from the ionization of acidic group. Now see, what is acidic ion exchange? So as we know, cationic exchanger has negatively charged beads are present in a resin. So this negative charge get from a functional group. For example, we have COOH, that is carboxylic acid group, as a functional group. If we remove one proton, it get COO minus. So this is how the beads get a negative charge. Example, purolite C100. Anionic exchanger. Similar, in anionic exchanger, it has positively charged groups that will attract negatively charged anions. These are called basic ion exchange. Now here beads are positively charged and this positive charge they also get from a functional group. That's why they call as basic ion exchange material. Example, DEA, that is dimethyl aminoethyl. Now see the diagram. In first diagram, anion exchanger stationary phase particle have positively charged cations which further attract negatively charged anions, and these anions are replaced with anions in a mobile phase, and finally the anion are eluted out. In second diagram, cationic exchanger stationary phase particle have negative charged, it means it will attract positively charged cations, and finally the cations are eluted out. Now what is anionic and cationic exchange? Cationic exchange, the separation of cation using cation exchange resin, as the cation to be separated are present in a given solution and exchange for similar ion present in a cation exchanger resin, that is solid matrix. For example, see here I have mentioned one equation, x plus a minus plus b plus, consider it as a stationary phase, and mobile phase c plus and d minus is cation or anion. So A is nothing but agarose, which is charged with a negative and it will attract of course positively charged cation. So here B plus is a cation which is further get exchanged with a C plus which is cation in a mobile phase. So this is how cationic exchange is done, and finally the cations are eluted from the column. Anionic exchange, the separation of anion using anion exchanger resin, as the anion to be separated are present in a solution and exchange for similar ion present in ion exchanger resin in a solid matrix, and finally anion eluted out of the particular column. Okay, first of all I'll explain this. An impure protein sample is loaded in a ion exchange chromatography in previous diagram. Positively charged protein molecule are bounded to the negatively charged group of resin molecule. A salt gradient, that is basic NaCl salt, is used to an elute to separate protein from resins. At a low salt gradient, a few positively charged protein molecule is eluted from the resin column. At a high salt gradient, most of the positively charged protein molecule is eluted from the resin column. Now see the diagram. First of all, we have to load the sample. In second step, you have see that positively charged ion protein with immobilized ion surface. In third column, eluted with low salt gradient, the few molecules are eluted. Or in fourth, eluted with high salt gradient, it means high NaCl is used, more chloride ions are get eluted from the column. Now how pH affect elution? The pH scale value is used to elute protein on the basis of its isoelectric point. See, the isoelectric point is point where H plus and OH minus concentration is similar. It means the net charge is 0. At an isoelectric point, amino acid in the protein molecule do not carry any charge and they do not migrate towards an electric field, as I mentioned. A decreasing pH gradient is used for the elution of protein from anion exchanger resin, and increased pH gradient is used to elution protein from cation exchange resin. Now, choices of buffer. What is buffer? So, buffer is nothing but a mixture of acid and its conjugate base. Basically, it is a salt. The decision include not just the buffer substance but also the pH and the ionic strain. Buffer ion will of course interact with the ion exchanger resins, will stop buffer ion with a charge opposite to that of ion exchanger, compete with solute for binding site of greatly reduce the capacity of column. Cationic buffer should be used with anionic buffer and vice versa. The pH chosen for the buffer depends upon all the ranges of the stability of the macromolecule to be separated. Second, the buffer pH should be chosen that the desired macromolecule will bind to the ion exchanger. In addition, the ionic strength should be relatively low to avoid damping of the interaction between solute and ion exchanger buffers. The buffer concentration, it may be in the range of 0.05 to zero point 0.1 molar is generally recommended. Now, classification of resin, generally based on chemical nature, it is of four types. Strong cation exchanger resin SO3H, weak cation exchanger resins have COH, OH and sulfhydryl group as a functional group. Strong anion exchanger resins have NR2, N plus R3, while weak anion exchange resins have NHR, NH2 and many more. What is working principle? How is it work? The charged molecule in sample are separated by electrostatic force of attraction when passes through an ionic resin at particular pH and temperature. It means pH and temperature is maintained throughout the process. The separation occur by reversibly exchange of ions between ion present in solution and those present in ion exchange resins. This form of chromatography relies on the attraction between oppositely charged stationary phase known as an ion exchanger and analyte. The ion exchanger basically contains charge group covalently linked to the surface of an insoluble matrix. The charge group of the matrix can be positively or negatively charged. When suspended in an aqueous solution, the charge group of the matrix will be surrounded by ions of an oppositely charged. This is how it works. In this ion cloud, ion can be reversibly exchanged without changing the nature of the property of the matrix. Now, part of instrument used in ion exchange chromatography. First, pump. The IC pump is considered to be one of the most important component in the system, which has to be provided continuous constant flow of the elutant through the ion exchange chromatography injector, column and detector. Now, what is injector? Sample introduction can be accomplished in various ways. The simplest method is to use the injection valve. Liquid sample may be injected directly and solid sample need only to be dissolved in an appropriate solvent. Injector should provide the possibility of injecting the liquid sample with the range of 0.1 to 100 ml of volume with high reproducibility under high pressure, which is up to 4000 psi. Now, columns. Depending on its ultimate use and area of application, the column material may be stainless steel, titanium, glass, or an inert plastic such as PIC. So, see, the column material is generally used, which has to be non-reactable. The column can vary in diameter about 2 mm to 5 cm and in length from 3 cm to 50 cm, depending on whether it is to be used for normal analytic purpose, microanalysis, high-speed analysis and preparatory work. Suppressor. The suppressor reduces the background conductivity of the chemical used to elute sample from the ion exchange column, which improves the conductivity measurement of the ion being tested. IC suppressor are membrane-based devices which are designed to convert the ionic eluent to water as a means of enhancing the sensitivity. Detector, generally electrical conductivity detector is used. Data system. In routine analysis where an automation is needed, a pre-programmed computing integrated may be sufficient. For higher control level, a more intelligent device is necessary such as data station and mini computers. Advantages and disadvantages. It is one of the most efficient method for the separation of charged particle. It can be used for almost any kind of charged molecule including large protein, small nucleotide or maybe amino acids. Ion exchange is used for both analytic and preparative purposes in laboratories, the analytic use being the more common. Inorganic ions also can be separated by ion exchange chromatography, so there are some limitations also. Only charged molecules can be separated, and buffer requirements. Um, what are the applications of ion exchange chromatography? Separation of ion from one another because different ions undergo exchange reaction to different extents, removal of interfering radicals, softening of hard water, complete demineralization of water, purification of organic compounds extracted in water, separation of sugar, and separation of amino acid. So, yeah, this is all about ion exchange chromatography. Thank you.